All right. So now we next move on to the another important component of the cell, which is the endomembrane system. The endomembrane system is a system made up of membrane-bound organelles, which are important for a variety of different functions. These organelles they contain a single membrane around them, and they contribute to various cellular processes. These include the endoplasmic reticulum, both smooth and rough, the Golgi complexes, and then the vesicles and other components which originate from the ER-Golgi system. This includes the lysosomes, peroxisomes, and other structures and other vesicles as well. So collectively, all of these components they make up the endomembrane system. So the endomembrane system it comprises of the endoplasmic reticulum, the Golgi complex, the vesicles which are produced, and then vacuoles and other related organelles, including the peroxisomes. So this figure here shows an overview of the various components of the endomembrane system. So this represents the nucleus, and here you can see that the endoplasmic reticulum are shown in this part of the cell. Well, actually, to simplify, the organelles are shown in only one particular region of the cell. Actually, they are spread throughout the cell. So the Golgi, so the ER, endoplasmic reticulum, they are not only associated with the nucleus, rather they will be present everywhere in the cell. So the endoplasmic reticulum are important for protein synthesis, for detoxification, for lipid synthesis, and so on. So the endoplasmic reticulum, um, there are two types of the ER. With some endoplasmic reticulum, ribosomes are attached. So you can see over here that these small circles are the ribosomes. So the ribosomes are attached to the endoplasmic reticulum here. So when they are observed under the microscope, their surface looks rough because of the attachment of the ribosomes. So these are called as the rough endoplasmic reticulum. Rough endoplasmic reticulum, they are important for the synthesis of proteins. So as the ribosomes make a protein, the protein it then passes through the membrane of the rough ER inside this cavity. The cavity of the endoplasmic reticulum is called as the lumen of the ER. So the proteins from the ribosomes are transferred into the lumen of the endoplasmic reticulum, and then they move through this lumen, and eventually it bud off in form of transport vesicles. So a portion of the membrane of the endoplasmic reticulum will bud off. So the membrane will start to fold inward, and then a portion of the membrane, along with the protein and other molecules entrapped inside, it will pinch off, and that is called as a transport vesicle. Those vesicles then transport these proteins and other molecules to the Golgi complex. The other type of the endoplasmic reticulum does not have any ribosomes attached to their surface. So when observed under a microscope, their surface would appear smooth. So therefore, that gives the name smooth endoplasmic reticulum. Smooth ER is not directly involved in protein synthesis, however, it carries out other functions. There are enzymes which are important for detoxification of harmful substances. This include a variety of different types of substances like the drugs, metabolites, other substances produced inside and coming from the outside as well. So they are modified by the enzymes which are present in the smooth endoplasmic reticulum. So therefore, it is important for detoxification and conversion of substances into other forms. Then the smooth endoplasmic reticulum is also important for lipid biosynthesis. So most of the lipid molecules for the membranes, they originate from the smooth endoplasmic reticulum. So these are the two important types of the endoplasmic reticulum, the smooth and the rough ER. So the transport vesicles or the transition vesicles, they bud and then they carry the cargo molecules with them. The cargo molecules include different types of proteins and other substances as well. Now, we know that proteins are produced by the ribosomes. So there are three ribosomes present in the cytoplasm of the cell. Those proteins which need to work inside the cytoplasm of the cell, they are produced by the free ribosomes. Whereas the proteins which need to go to a certain destination, they are produced by the rough endoplasmic reticulum. So these ribosomes are attached to the ER, then these proteins are transferred into the lumen of the endoplasmic reticulum, then through the transition vesicles, the protein molecules are transferred to the Golgi complex. So in the Golgi complex, the membrane of the vesicle fuses with the membrane of the Golgi. As these vesicles they fuse with the Golgi, so their cargo molecules they are transferred to the lumen of the Golgi complex, and then these molecules they undergo processing, they undergo modifications. Many proteins they undergo modification after synthesis. This process of protein synthesis is called as translation. So these proteins they undergo modifications. These are called as post-translational modification. Post-translational means that after translation has occurred, protein synthesis has occurred, then these proteins they undergo modification. There are different types of modifications which occur on these proteins. One common type of modification is glycosylation, which is the attachment of carbohydrates. Different types of carbohydrate molecules, including several different types of oligosaccharides, they are attached to the proteins. And this attachment of the carbohydrates to the proteins mainly take place inside the Golgi complex. Then proteins they undergo other types of modifications as well. So these post-translational modifications, most of them, they occur inside the Golgi complex. Some modifications do take place in the ER, but most of the modifications of the proteins, they occur inside the Golgi complex. This is sometimes also called as the ER-Golgi pathway, where the modification of the proteins takes place. So those proteins which have to go to a certain destination besides the cytoplasm of the cell, they are transported through the ER-Golgi pathway. So inside the Golgi, the proteins undergo modification, and as they are modified, then the Golgi sort of like process the proteins and package them into these transport vesicles. The transport vesicles then carry the proteins to their destination. Some of the proteins, they are packed in the vesicles which are called as the secretory vesicles. The secretory vesicles are important for secretion of the protein. So the protein molecules or other different types of cargo molecules like carbohydrates and lipids and others. So all those molecules which are to be secreted, like hormones and so on, so they are packaged inside the secretory vesicles. And then the secretory vesicles they fuse with the membrane. So as they fuse with the plasma membrane of the cell, these molecules are exported out of the cell, resulting in a cell secretion. The other uh, fate of the vesicles is that some of the proteins they need to work at other cellular locations. For example, some proteins need to work in the plasma membrane, inside an organelle membrane, or in the nucleus, in the membrane of the nucleus, in mitochondria or other organelles, in a matrix, in the membranes. So as the vesicles they go to their destination, they would deliver the proteins to their target. Now, how is it decided that to what destination this vesicle is going to transport the proteins? That actually is decided through a mechanism of tagging. All the proteins which need to go to a certain destination, they're processed in a particular manner, and then they can have sort of like a tag or an identifier that is going to describe the destination of the protein. So that tag or that identifier is used to this this more like a postcode which is going to identify where this protein is going to go. So based on those tags, on those identifiers, then the proteins are transported to their given destination. So in this manner, the transport vesicles would transport the protein to their destination, which could be like secretory vesicles transport outside the cell, or the transport vesicles would transfer the protein to the destination like the membrane or other organelles, organelle membrane, or the matrix or the stroma of the organelles. So in this manner, the proteins are transported. Also, the Golgi complex process a group of proteins which are called as acid hydrolases. The acid hydrolases the acid hydrolases are hydrolytic proteins. These are enzymes which carry out hydrolysis of the molecules. They include proteases, nucleases, lipases, carbohydrate metabolizing enzymes. So all of these different types of enzymes, since these enzymes they work at an acidic pH, so therefore collectively these are called as acid hydrolases. So the Golgi also processes these enzymes, the acid hydrolases, and then the vesicles containing the acid hydrolases diffuse with other uh, vesicles. For example, one way of producing the vesicles is through endocytosis. So, external material, well, it could be like food material, it could be like small particles or aggregates, it could be bacterial cells or other smaller cells which are engulfed by a cell. So, as they enter the cell through endocytosis, they will be wrapped around by a small portion of the plasma membrane of the cell. This is called as an endocytic vesicle. So the endocytic vesicle then develops into an endosome. So these transport vesicles, which carry the acid hydrolases, they fuse with the endosomes. And as they fuse with the endosomes, their acid hydrolases, their enzymes are poured into the endosome. And then these hydrolases, they will start breaking down the material present inside the endosome. This then develops into a lysosome. So lysosomes, they contain acid hydrolases and they are important for the breakdown, degradation, and of different types of substances. So then there is another important organ that's called as the peroxisome. We would look at the peroxisome and their function later on. So collectively, all of uh, this system which is made up of the endoplasmic reticulum, Golgi, lysosomes, peroxisomes, and then different types of vesicles. Collectively, this entire system is called as the endomembrane system. The components of the endomembrane system, they are surrounded by a single membrane. So now we are going to look at these different constituents of the endomembrane system one by one. So the first important constituent is the endoplasmic reticulum. So endoplasmic reticulum is a continuous network of tube-like structures, and they are on one side, they are continuous with the nucleus. So if you would see here, looking at the figure again, so you can see that the membrane of the endoplasmic reticulum is continuous with the nuclear membrane. So on one side, it is continuous to the membrane, and on the other side, via the Golgi and vesicles, it is continuous to the plasma membrane. So the endoplasmic reticulum is actually like continuous with different components of the cell. So so endoplasmic reticulum, that means inside the cytoplasm. So a network present inside the cytoplasm. So this is the literal meaning of endoplasmic reticulum. So there are membrane-bound sac-like structures in the endoplasmic reticulum, which are called as the cisternae. So cisternae are so these sac-like structures, these regions, folded sac-like structures, these are the cisternae of the endoplasmic reticulum. And the interior, the cavity inside the cisternae, is called as the lumen of the endoplasmic reticulum. So yeah, this is another diagrammatic representation. So you see this is the nucleus, and the membrane of the nucleus is continuous with the endoplasmic reticulum. So in this part, the rough endoplasmic reticulum are shown, which are bound by the ribosomes, and then here are the smooth endoplasmic reticulum. So this is a micrograph showing the structure of the endoplasmic reticulum. So if you look here on the left panel, so you can see that there are these small dots present on the surface of these sac-like structures. So these are the ribosomes which are attached onto the surface of the endoplasmic reticulum, thereby producing the rough ER. As compared to the rough ER, if you would look at the right panel, so these are the smooth ER. So you can see that these are the sac-like structures of the ER, and they do not have any ribosomes attached to their surface. Thereby, their surface appears smooth under the microscope. So the region of the now, what happens is that so if this is representing a region of the endoplasmic reticulum. So it happens that the cargo molecules they're accumulated, and then the molecules are going to accumulate in the terminal part of the endoplasmic reticulum. And then this terminal part, the membrane will start to fold inside, and therefore the membrane is going to fuse here. Thereby, this portion of the endoplasmic reticulum will be released in form of a vesicle containing the cargo molecules in it. So in this manner, this portion will be separated. So these regions of the endoplasmic reticulum where the which are important for sort of like budding off. These are called as the transitional elements, and they are important for producing the transition vesicles. So when the vesicles will be produced, the vesicles will then go to mainly the Golgi, and therefore the membrane lipids, the lipids present inside, the proteins, carbohydrates, and other molecules which it is carrying. So all those molecules will then be transported to the Golgi, and then from the Golgi, they will be channeled to their destination. So in this manner, the vesicles, they bud off from the air, and then they transport the carbon molecules to the Golgi. Now, how it is decided that which proteins need to go to the endoplasmic reticulum for synthesis. So the cell contains free ribosomes present in the cytoplasm. Those proteins which need to work in the cytoplasm, so their messenger RNA will be bound by the ribosome, and these free ribosomes will make a protein, and then the protein is going to stay inside the cytoplasm of the cell. However, those proteins which need to go to a certain destination, they are produced by the attached ribosomes. Initially, what happens is that these proteins, so when a protein synthesis is going to start, so okay, so if this represents the ribosome, this is the large subunit of the ribosome, and this is a small subunit of the ribosome. So the red colored structure is the messenger RNA, and the green colored thing is the protein. So as the ribosome would start translating a protein, then the initial few amino acids of the protein are produced. If it is actually this portion of the protein. This is the N-terminal part of the protein, which contains a specific sequence of amino acids on it. That is called as a signal peptide. The signal peptide is about like 20, 30 amino acids, about that much. So, and it is present on the N-terminal. So the first few amino acids which are translated, they contain sort of like a code that where this protein has to go. So the destination of the protein is sort of like coded in the sequence of the amino acids. For every particular destination, there is a unique signal peptide. That signal peptide is going to direct the protein to its destination. So this about like 20-ish amino acids on the N-terminus, which are produced first. So that is sort of like the address of the destination for this particular protein. If a protein does not contain any signal peptide, then the protein synthesis will keep on continuing in a normal fashion, and the ribosome will keep on moving, therefore keep on producing the protein. And once protein synthesis is complete, then the protein will be released in the cytoplasm of the cell. So the protein will stay in the cytoplasm. These are called as soluble proteins. They're going to stay here inside the cytoplasm and they're going to work here. But if a protein needs to go to a destination like a membrane, like an organelle, or like outside the cell, then the protein will contain a signal peptide on it. And that signal peptide will be a sequence, a unique sequence of amino acids present on the N-terminus. So if a protein does not contain a signal peptide, it's going to be cytoplasmic protein. But if a signal peptide is present, then that signal peptide will be recognized by a machinery in the cell. There is a complex in the cell that is called as the signal recognition particle, or abbreviated as SRP. The SRP is a complex which would sort of like scan the newly synthesized proteins and look for a potential signal peptide. So if a signal peptide is detected, then the SRP is going to bind to the complex and it is going to pause the process of protein synthesis. And then the SRP is going to carry the entire complex of the ribosome with the messenger RNA and all other cofactors with it. It is going to migrate all of this entire complex to the membrane of the endoplasmic reticulum and attach there. So the ribosomes, when they start producing a protein, they would initiate the process of protein synthesis as free ribosomes. It is later that they will be transferred by the SRP to the membrane of the endoplasmic reticulum. This is co-translational translocation. So there is the protein is going to be translocated, it's going to be migrated into the lumen of the endoplasmic reticulum, and this process of translocation takes place while the process of translation is taking place. So as the protein is getting synthesized, it will be translocated inside the endoplasmic reticulum. So this is called as co-translational translocation. Protein is going to be translocated, it's going to migrate, and this migration is co-translational. That is, it is along with the synthesis. In most cases, when a protein has been synthesized, this signal peptide is no longer needed because the protein has been identified to go to a certain destination. So now the signal peptide is no longer needed. Hence, an enzyme present in the endoplasmic reticulum called as signal peptidase. The enzyme peptidase will remove the signal peptide, and therefore the protein will be transferred from the endoplasmic reticulum to the Golgi and then will go to its destination. So the process is represented here in this diagram. So this is the messenger RNA shown in orange color. The transfer RNA is represented in yellow, and then this is the ribosome shown um, in cyan color here. So, as the ribosome will start making a protein, if the protein contains a signal peptide on it, that will be recognized by this complex called as the signal recognition particle. So, SRP would recognize a signal sequence and then it will stall, it will pause protein synthesis here. The SRP has a receptor called as the SRP receptor present on the membrane of the ER. So the SRP will translocate, will carry the entire complex to its receptor and bind here. Associated with a membrane is a translocation pole. So as the SRP would bring the ribosome here, then this would be followed by GTP hydrolysis, and then the energy from GTP hydrolysis will be used to sort of like push the newly synthesized portion of the protein into the channel. So the channel will open and then the protein will be attached here. So in this manner, the ribosome will now start translating the protein. And as it would read through the sequence of the messenger, it will produce the protein, and the protein through this channel is going to be translocated inside the membrane, through the membrane of the ear into the lumen. So the protein synthesis will start. Then there is this enzyme called signal peptidase. The signal peptidase enzyme will recognize the signal sequence and then it is going to hydrolyze the peptide bond here. So the ribosome will keep on synthesizing the protein and the protein will then be translocated through the channel inside the lumen of the endoplasmic reticulum. So that is how the proteins, they will end up inside the ER lumen. Then through the transition vesicles, these proteins, they will be packaged and delivered to the Golgi for further processing and delivery to the destination. So one important aspect is attachment of carbohydrates to the protein. These proteins are called as glycoproteins because they contain carbohydrates attached to them, and these carbohydrates are important for various functions. One function of the carbohydrate is that carbohydrates are attached in the ER and mostly in the Golgi. So these carbohydrates which are attached, number one, they act as an identifying marker for the destination of the protein. So if a wrong carbohydrate is attached, the protein can be sent to a different destination. So one function of these carbohydrates is that they are important for identifying and sending the protein to their destination, like a tag. Then the carbohydrates which are attached to the protein, they are important for stabilizing the protein structure. They are important for the protein function as well. So therefore, this process is called as glycosylation. Attachment of carbohydrates to the proteins is called protein glycosylation. And this process is important for proper folding and function of the proteins. Then the proteins are sent to the Golgi. Then in addition, some of the proteins, they need to form disulfide bonds. So the disulfide bonds are formed between the sulfur atoms of the amino acid cysteine. So cysteine contains sulfur atoms in it, and these sulfur atoms, they will attach to each other to produce a disulfide bond. So those proteins where disulfide bonds are to be formed, they are also transferred inside the lumen of the endoplasmic reticulum. So the ER contains this enzyme that is called as protein disulfide isomerase. So this enzyme is also present in the lumen of the endoplasmic reticulum. So the formation of disulfide bonds also take place inside the lumen of the endoplasmic reticulum. So all of these different types of modifications they occur. So like for example, formation of disulfide bond, glycosylation, which is important for the sending the protein to their destination. And then this is also important for the structure and function of the proteins. So all of these things, they take place, they're initiated in the endoplasmic reticulum, and the process continues inside the Golgi as well. Another important thing for the ER is that there are proteins which are improperly folded, or the proteins which are no longer needed. So the cells, they contain a complex that is called as a proteasome. Proteasome is important for the degradation of the proteins. Those proteins which are not required, either these are like sort of old molecule of the proteins which need to be degraded, or sometimes the proteins are misfolded, they are not functioning properly. So those protein molecules, they need to they need to be destroyed because they are no longer needed or they are non-functional. So therefore, all those molecules of the protein which are to be degraded, they're sent to the proteasome. The proteasome is a complex in the cell that is important for the degradation of the proteins. So the proteins, so ER is also a site of the quality control. So if proteins are improperly folded, they are misfolded, or they are not properly assembled, so then they are exported from the ER to degradation by the proteasome. This process is called as ER-associated degradation. So if the process does not take place properly, then misfolded and improperly formed proteins that will accumulate in the cell, and they can lead to diseases like cystic fibrosis, hypercholesteremia, and other diseases as well. Then a smooth endoplasmic reticulum, one important function is that smooth ER contains a group of enzymes which are called as cytochrome P450. Cytochrome P450 is a large group of proteins, and these proteins are important for the process of hydroxylation. Hydroxylation is attachment of hydroxyl groups to the molecules. So this is hydroxylation is mediated and attachment because hydroxyls are polar groups. So they increase the water solubility, they increase the polar nature of the molecules. They make substances more soluble, and then many of the hydroxylation reactions, they are important for formation of useful substances in the cells. So, for example, like many of the hormones or other molecules which are needed by the cell, they need to undergo hydroxylation. So that hydroxylation is mediated by the smooth endoplasmic reticulum. Also, the SCR, the cytochrome P450 group of proteins, they are important for hydroxylation of many of the drugs, xenobiotics, and other molecules as well. They also are important for hydroxylating the harmful substances. Therefore, they make them water soluble, easy to excrete, and therefore contribute to detoxification. Then the ER, the smooth ER, is also important for carbohydrate metabolism. So you see that cells like liver cells, they store glycogen in them. There's this enzyme called glycogen phosphorylase. The enzyme, it will phosphorylate glucose and release it. It will break down starch, break down glycogen, and is going to release glucose in form of glucose 1-phosphate. This will then undergo isomerization reaction to produce glucose 6-phosphate. So since the liver cells right now, they're going to release glucose in cases when when the cells, they require glucose. So the glycogen stored inside the liver cells will be hydrolyzed, and then the glucose will be released in blood. Say for example, like in response to glucagon, the glycogen breakdown will be stimulated. So as this glycogen is broken down, it will be converted into glucose 6-phosphate. Now, glucose 6-phosphate, its permeability through the membrane is quite low because this is a highly polar molecule and it contains a phosphate group attached to it, which is negatively charged. So this is a highly polar molecule. So in order to transport glucose, so this first must be dephosphorylated. Dephosphorylation means removal of the phosphate group. Phosphorylation is the attachment of phosphate, and dephosphorylation is the removal of the phosphate. So the dephosphorylation, the enzyme glucose 6-phosphatase. Phosphatases are enzymes which break down which release the phosphate group. So the phosphate group will be removed, and the hydrolysis would produce glucose. And then this glucose can be exported out of the cell through the glucose transporter. So the enzyme, which is important for removal of the phosphate, thereby making glucose permeable, is localized on the smooth ER. Then the smooth ER is also important for storage of calcium. The concentration of calcium inside the cytoplasm of the cell is typically low. Most of the calcium in the cell is stored inside the endoplasmic reticulum and the mitochondria. So the you would perhaps remember uh, the the process of muscle contraction from your high school biology class, that the endoplasmic reticulum of the muscle cells, well, that's called as the sarcoplasmic reticulum. It houses calcium in it, and when stimulated, calcium is released, and that is required for the movement, the contraction of the muscle fibers. So in this manner, the muscle contraction is dependent on calcium. Well, there is one role of calcium. Calcium is important for many other processes as well. So there are these calcium pumps called as the ATP-dependent calcium uh, ATPases. So these pumps, they use the energy of ATP hydrolysis, and they transport the calcium ions into the ER. So when the cell requires the ER to release calcium, so through a signaling mechanism, calcium channels are activated, and then calcium ions will be released into the cytoplasm of the cell. One function is the muscle contraction. Well, release of calcium is important for other signaling processes as well. Details are not important here, but this is just to show that there's a receptor in the membrane the ligand, like a hormone or other substances, they bind and stimulate the receptor. When the receptor is activated, it's going to convert a membrane phospholipid. So membrane phospholipid will be hydrolyzed, and then this molecule, this is inositol, shown in green color. So this molecule will be hydrolyzed and released in the cytoplasm of the cell. This is called as inositol triphosphate. So inositol triphosphate, uh, so this green colored um, thing, which is actually inositol triphosphate, is released. It is actually a part of the polar head group of the phospholipids. So the receptor will be activated. It will activate another phospholipase. The phospholipase will break down the bond and release inositol triphosphate, which was actually a component of the polar head group of this lipid. So this will be released into the cell, and then it is going to bind to the channels on the membrane of the ER, and therefore calcium will be released from the ER. So this calcium which is released, it then carries out other processes. Under the conditions when the cell does not require high amount of calcium in the cytoplasm, so there are proton pumps. The pumps are going to push calcium back inside the ER. So okay, so yeah, this is a similar thing that the membrane uh, of the ER. So the calcium ions are stored there, and when stimulated, calcium ions will be released from the ER into the cytoplasm of the cell. So this is important for muscle contraction because we know that there are the myosin motor proteins associated with actin cytoskeletal filaments, and they are important for muscle contraction. Then calcium also acts as a cofactor for many different types of enzymes. Calcium is also required for cell secretion when the vesicles are carrying the cargo molecules are to be transported. So that is mediated by the motor proteins like myosin. So the myosin on one side they bind to, let's say, like, okay, so if this is representing the actin filament, and this is a myosin shown in red color, and then this is a transport vesicle containing the cargo molecules in it. So the myosin motor proteins, they actually like carry, they bind to the cytoskeleton on one side, and they sort of carry the cargo, the vesicles with them. So as the motor protein, it sort of slides along the actin filament, it will carry the vesicle with it. So in this manner, they will carry and okay, so this is the plasma membrane here. So the motor protein will bring the cargo molecule here, and then the vesicle, it will fuse with the membrane, and therefore the substances, they will be secreted out of the cell. So in this manner, release of calcium is also important for cell secretion. Then there is another important protein in the cell that is called as calmodulin. This is a calcium binding protein, and calmodulin is important for various other physiological functions. It is important for regulating energy metabolism. It's important for cell signaling and other processes as well. So the release of calcium is actually going to perform a variety of different functions in the cells. And calcium is stored in the smooth endoplasmic reticulum. And the smooth ER is also important for steroid biosynthesis. So the different types of hormones which are derived from cholesterol. So steroids are actually hormones which are derived from cholesterol. So cortisol, testosterone, estrogen, and others. So these are all produced in the smooth endoplasmic reticulum. Then the smooth ER are also associated with plastids and plants, and then they're important for the phytohormone synthesis. Another important thing is the synthesis of cholesterol. So it's a complex pathway involving many enzymes. So one of the important enzyme that is called as HMG-CoA reductase. This is a committed and the most important step of the pathway of cholesterol biosynthesis. And this enzyme uh, is a site of regulation by drugs. So the cholesterol lowering drugs, these are called as statins. So the cholesterol lowering drugs, they actually bind and inhibit this enzyme HMG-CoA reductase. Then a smooth ER is also, well, both smooth and rough ER, they're important for synthesis of the membrane. So the smooth ER makes cholesterol, steroids, phospholipids, and then these lipid molecules through the shuttle vesicles move to the Golgi and to other components. So therefore, there is a continuous cycling of the membrane throughout the cell. So in this manner, these are important for the ER is important for membrane biosynthesis. And as the lipids are synthesized, so there are these translocators called as the flipases, so they can move the lipids from one leaflet to the other. In this manner, the membrane portions of the membrane are synthesized by the ER, and through the vesicles, they are transported to other components of the membrane, including the Golgi membrane, organelle membrane, and then eventually the plasma membrane of the cell. The next component of the endomembrane system are the Golgi. So the Golgi are okay. The Golgi are also flattened, sort of like small sacs of the membrane, and so the region of the Golgi which is closer to the ER, that is called as the that is called as the cis Golgi network. So the vesicles. So they move from the ER and then they are transported to the Golgi. So the region of the Golgi close to the ER, which is going to receive the vesicles, that is called as the cis Golgi complex or the cis Golgi network. So the transition vesicles are going to fuse here, and then the cargo molecules they move from these uh, cisternae of the Golgi. So these are the medial cisternae, and then through these cisternae, the cargo molecules are transported to the opposite phase. This is called as the trans-Golgi network. And then the vesicles they bud off from the trans-Golgi, and they are going to transport the vesicles to their destination. This is a micrograph showing the Golgi. So this is a cis Golgi network, and this is the trans Golgi network. So carbohydrates, one major modification is glycosylation. There are actually two types of glycosylation. In one type of glycosylation, the carbohydrates are attached to a nitrogen atom of the protein. This is called as N-link glycosylation. And in the other, the carbohydrates are attached to an oxygen atom of the amino acids. This is called as O-link glycosylation. Well, all the details are not important here, but this figure is to show the various changes which are brought about through the ER-Golgi pathway. So in the ER, the base oligosaccharide is attached, and then the initial processing is done. Then this is transferred to through the vesicles to the cis Golgi, and then through the cis Golgi transferred to the medial, and then to the trans Golgi. So different types of carbohydrates are attached, like for example, O-linked, N-linked glycosylation. Um, and O-linked glycosylation continues here. For example, attachment of N-acetylgalactosamine, which is an amino sugar that is attached here. Then mannose are removed phosphorylation of the proteins is done. Then further modification of the oligosaccharides are done, like attachment of galactose, attachment of sialic acid, attachment of sulfates. So all these modifications, they continue through the passage of the proteins through the Golgi. And eventually the modified proteins, they bud off as vesicles and then transported to their destination. So there are two important cycles of movement in a Golgi. So these are called as the anterograde and retrograde. If I was sure. Okay. So the anterograde movement, it transports the molecules from the ER Golgi to the membrane, plasma membrane, and the retrograde is the backward movement. So this is the ER, the Golgi. So molecules, proteins, and carbohydrates, lipids from the ER go to the Golgi. Then from the Golgi, they go to the membrane or other destinations. So this is the anterograde, which is a forward movement. And then the vesicles, they can sort of like go back to the Golgi, and but from Golgi go back to the ER. This is the retrograde transport. So the process of movement is shown here, that this is a rough ER, and then there are smooth ER. So the proteins, lipids, and carbohydrates, so they're packaged into vesicles. These transition vesicles then fuse with the on the cis Golgi network, and then the modifications are done, and these proteins, they undergo these modifications, and then eventually they product from the trans Golgi. So if the protein is to be secreted out of the cell, then it is packaged into these vesicles called secretory vesicles, and exocytosis takes place. Or the vesicles, they can transport the molecules to other cellular destinations as well. So molecules end up in the cell through endocytosis as an endosome, and then the hydrolytic enzymes are transported to these endosomes, fuse with them, and therefore turn them into lysosomes. So this figure is showing the mechanism of trafficking through the endomembrane system. This is sometimes also called as the ER-Golgi pathway. Now, some proteins, they need to stay in the ER, or they need to stay inside the Golgi. So these proteins, they contain ER specific retention or retrieval tags. So if a protein has to reside in the, like, has to work inside the ER, so these resident proteins, they should be retained inside the ER. So they contain sort of like a retrieval tag on them, or a retention tag. So this is a specific sequence of amino acids on the protein which help in retaining the proteins inside the endoplasmic reticulum. So in this manner, these proteins, the ER proteins, they do not go to other destinations. They are retrieved back to the endoplasmic reticulum. So how do the proteins, while they're moving through the Golgi, they undergo all these modifications? Well, there are two models that two possible explanations for the modifications of the proteins as they go through the Golgi. The first model is the uh, stationary cisternae model. In this model, the cisternae, they are okay. So you see that different modifications, they take place in different regions of the Golgi. So the stationary cisternae model states that the cisternae, they are isolated from each other, and the vesicles bud off from one cisternae, carrying the molecules and fuse with the next one. Then some modifications takes place here, vesicles bud off, and then they go to the next cistern. In this manner, the vesicles keep on budding off and carrying the modified proteins with them to the terminal pod, and then eventually the vesicles are released. So this is called as the stationary cisternae model. The other model is the cisternal maturation model. This model states that the cisternae, they are under they're dynamic, so they are under continuous remodeling. So as let's say, okay, so let's say these are the cisternae. the Golgi. So initially, the proteins would be received here. So this will be the cis Golgi, and the proteins are like sort of they arrive here. And when some set of modifications have been done, then this will accumulate the enzymes which are required for the next round of modification. So now the same cisternae will develop into the uh, medial cistern. And then later those modifications have been done, then the same compartment will accumulate more enzymes in it, which will be required for the next round of modifications. Hence, this will turn into trans uh, Golgi membrane. So, in this manner, the cisternae are under continuous remodeling by accumulating specific type of enzymes which are required for a certain type of a modification.